So I'll start by thanking Hugues for the kind invitation. It's certainly a privilege and an honor to speak here about our work. So I'm going to tell you experiments that we did in our lab that led us to actually conclude that P53 is a major regulator of telomere metabolism. So this work was done mainly by two PhD students in the lab. It was done mainly by Eva Simonova, a former PhD student, and then by Sarah Jaber, another PhD student in the lab. And they were helped by Boris Bardot, Rachida Bouariche, Ming Fang, and Vincent Lejour. And we were fortunate to have neighbors that were expert in telomeres, and we had a great collaboration with them, especially, so the lab is Arturo Londono's lab, and especially we worked with Elena Draskovic and David Silvestre. So Laura told you a lot about the N-terminus of the protein, of the P53 protein, and we were actually interested in the other end of the protein, the C-terminus. And as you can see here, the C-terminus is the site of many post-translational modification. You have methylation, ubiquitination, nedylation, sumoylation, acetylation, and phosphorylation. So this tells you that it's a very important domain for the regulation of P53 stability and activity. But what was a little bit confusing in the field is that these modifications could have opposite effects. So it was a bit difficult to guess what would happen if you actually remove this feature. And we were interested in doing that in vivo to try to understand what was going on really. So what we did is that we engineered a nonsense mutation here that would remove the last 31 amino acids of the protein, so basically the entire C-terminal domain. So we use classical homologous recombination to target this mutation in mouse ES cells, and then we derived mouse embryonic fibroblasts, MEFs, and you can see here what happens in these MEFs compared to wild type. So in unstressed cells, you can see that the P53 delta 31 protein is more abundant than the P53 wild type. And if you treat cells with adriamycin, you induce a DNA damage, you have accumulation of the wild type protein and also accumulation of the mutant, but this mutant is actually more abundant than the wild type. Then if you look at, so P53 is a transactivator, and if you look at the transactivation of classical target sites like P21 or MDM2, you can see that the mutant cells actually transactivate P21 more efficiently than wild-type cells. And you can see that in unstressed cells, in cells with DNA damage, and cells treated with nuclein, which is a specific P53 activator. And here this Western is actually confirming what I'm saying here with immunofluorescence and with qPCRs. Now we ask about the P53 P53 dependent cellular responses, and you can see here that if we irradiate MEFs with various doses of irradiation, you have a change in the G1S ratio, which is typical of P53 response that arrests cells in G1. So you see this G1S ratio changing in the wild-type cells, but it's actually even more dramatic in the mutant cells, indicating increased activity. We also looked at the proliferation of the cells. And you can see that in, so this is a 3T3 protocol, and you can see with this protocol that basically wild-type cells enter into a plateau, into senescence after 8 passages, whereas you have actually this plateau in like 4 or 5 passages in the mutant. So it's, again, increased activity, and we could see also beta-gal staining that was typical of senescence. Now, what about the mice? So we crossed together P53 heterozygous mice, and we got perfectly, well, almost perfect Mendelian proportion of the mutants. But the mouse, the homozygous mice, were actually much smaller than the littermates, and they had increased skin pigmentation at the foot pads, And if we looked at the thymuses, we could see, actually you can see here cellular fragmentation in the thymus sections stained by H&E, and you can see that there's actually spontaneous apoptosis in these mutants. So all this was actually demonstrative of increased p53 activity in different tissues. Now I told you that these mice are born in Mendelian proportions, but when we look at weaning, so 3 weeks later, there's a large underrepresentation of these mice. And this is due to the fact that most mutant mice, homozygous mice, die between 14 and 43 days after birth. So what do they die of? We dissected the mice, and we found that they have a much larger heart. Remember that these are much smaller than their littermates, yet they have larger hearts than their age-matched white-type counterparts. And here you can see that if you consider also the total body weight. So it's been described that enlarged hearts are actually indicative of possible anemia. And so we looked at blood counts. So we counted white blood cells, red blood cells, and platelets. And this is a comparison of 5 mutant mice and 5 wild-type mice. You can see that for the wild-type mice, all the values are within the normal range, whereas all the values are actually aberrant and very much lower than normal in the mutant. So this actually indicated not only anemia, but pancytopenia, and this indicated bone marrow failure. And indeed, you can see, so we did many, many different assays, but here you can see, for example, a section of the sternum, and you can see that there's a lot of bone marrow cells in the sternum of Y-type mice, and almost no cells in the mutant. So basically, these mice die of bone marrow failure very early on. And what was interesting is that they also developed pulmonary fibrosis. So this is a trichrome Masson staining that allows you to actually stain collagen in green, and you can see that there's a lot more collagen in the lung of the mutant compared to the wild-type mouse. And this was particularly interesting because in human, it has been shown that the combination of bone marrow failure and lung fibrosis is a hallmark of telomere dysfunction. So telomeres are the tips of the chromosome that protect actually the genetic material from being lost after successive generations. So we looked at the size of the telomeres in the mutant, and so that's where we started the collaboration with our neighbors, and we used a telomeric probe to detect telomeres, and with this probe, basically, I hope you can see that the intensity of the fluorescence is dependent on the length of the telomeres, and I hope you can see that there's an overall lower intensity of fluorescence in this cell compared to that cell. That's wild type. So we did that with many metaphases, and we could see really a shift towards a smaller telomere length. We also did that in bone marrow cells, and it's the same principle. Here, the fluorescence leads to a shift to the right, and you can see that this shift to the right is smaller in the mutant compared to the wild-type cells. And we could measure precisely the size of the telomeres. So it's not only telomere length, but it's also telomere dysfunction, and this is shown by the fact that we could see in the mutant cells a colocalization of a telomere signal with a DNA damage signal that's gamma H2AX foci. So this led to conclude that the p53 delta 31 mice, they model telomere syndrome. So the classical telomere syndrome is dyskeratosis congenita, and the classical physical traits that dyskeratosis congenita patients have is skin hyperpigmentation. I showed you that before. Nail dystrophy, which we could see in some mice, and oral leukoplakia, which is an aberrant structure of the mucosa. And you can see here tongue sections where there's an aberrant structure in the tongue of the mutant. Now, the pathological traits are bone marrow failure and pulmonary fibrosis, and I showed that to you before. And the molecular feature is poor telomere maintenance, and I showed that to you also. Now, what was interesting is that beyond these typical DC traits, you have also traits that are typical of people with severe dyskeratosis congenita and more particularly the Orier-Alredorsson syndrome. So these patients, in addition to all these traits, they have short stature, which obviously we saw in our mice, testicular atrophy, and we also saw testicular atrophy and arrested spermatogenesis in our mice, and cerebellar hypoplasia, and we also could see reduced size in cerebellum in our mutants. Now, this suggested that p53 was really important in regulating maybe different genes involved in telomere metabolism. And here you have basically a cartoon that shows you a little bit about these genes these proteins that are important for telomere metabolism. So the telomerase holoenzyme is the complex that will generate the TTA-GGG DNA repeats. And this holoenzyme is composed of a reverse transcriptase, RNA component, that's the template for the DNA repeats, and then accessory proteins, NOP10, NHP2, discarin, and GAR1. This complex is actually assembled in Cajal body, and TCAB1 is important for this assembly. And then you also have the CST complex and RTAL1 that are that are important for the synthesis and the maintenance of telomere length. And also, in addition to that, you have a structural component, which is the sheltering complex. It's a complex that will bind and protect telomeres, and it's composed of TIN2, RAP1, TRF1, TRF2, TPP1, and POT1. Now, from all these components, those in blue were shown to be mutated in dyskeratosis congenita, and one was particularly interested. It seemed particularly interesting. It was TCAB1, because the other name of TCAB1 is RAP-P53. It's a gene that's partially antisense to P53. So we thought that maybe we had a misregulation of TCAB1. But actually, when we looked at the expression of this gene in knockout white type or delta 31 cells, we didn't see a very significant difference. So it was actually not the gene that was responsible for what we were seeing. So we asked, does P53 regulate any of the 8 other genes? And we actually added another one, which is TRF1, which is not shown to be mutated in dyskeratosis congenita, but it had been implicated in aplastic anemia, which is a milder form of telomere syndrome. And you can see here the results. Actually, we found that 4 of these genes are downregulated in the delta 31 mice. So it's DKC1, discarin, RTAL1, TIN2, and TRF1. And for 2 of these 4 genes, we had access to good antibodies, and you can see that we see significant differences also at the protein level for RTAL1 and discarin. And then we used Nutlin to specifically activate P53, and you can see that clearly Nutlin leads to downregulation of these 4 genes in wild type of delta 31 cells, but not in the P53 knockout cells, which is a clear demonstration that this is P53 dependent. So as Laura told you, P53 regulates hundreds of genes, so how could we be so sure that by regulating these 4 genes we really impacted on the phenotype of our mice? And we actually got an information on that by the fact that our cohort of mice was heterogeneous in genetic background. So this cohort was 75% black 6 and 25% 129. So the black 6 mice have a black color coat, and 129 have a brown agouti color coat. And what we had noticed is that those mice here that survive longer than 90 days actually were all brown. So this suggested that so the color code is different due to an agouti locus on chromosome 2 of the mouse. And this suggested that maybe there was a gene close to the agouti gene that was important for the phenotype and the survival of our mice. And a good candidate was RTAL1 because it mapped 26 centimorgan away from agouti. Now we used a SNP in intron 15 of RTAL1 that allowed us to make a difference between a black6 and a 129 allele. And we generated more mice. And then we looked at the survival of these mice depending on the RTAL1 allele that they had. And you can see that those mice that had 2 black6 alleles were actually dying faster than those that had 1 or 2 129 alleles. So it was a very good indication. And also, when we looked at the bone marrow cells of pure black6 or pure 129 cells, it is it is clear that you have actually more RTAL1 expressed in the BMCs of 129 than of black6 mice. So this is a clear indication that at least for one of the 4 genes that we saw regulated by P53, the level of expression of this gene is very important for the survival of our mice. So this led us to this kind of cartoon, where if you have a mouse that's expressing wild-type P53, you have a normal P53 activity in a healthy mouse. Now, most mutants that have been studied so far are mutants that affect the core DNA binding domain of P53, which leads to a decreased P53 activity, a mouse model of Leaf-Romany syndrome, a cancer predisposition syndrome. And here we were actually seeing a germline mutation that removed the CTER of P53. This increases P53 activity, leads to a mouse model of a telomere syndrome and to aplastic anemia and lung fibrosis. Okay, so I told you only about mice. So, of course, a valid question is this relevant to human disease in any way? So I'll give you 5 points that make it for us a really valid and relevant to human disease. The first thing is that the P53 C-terminus is highly conserved between human and mouse. If you look at the last 31 amino acids, there's 81% identity in the last 31 amino acids. So it's kind of a weak argument, but you can guess that they might have similar function. The second point is that we actually used a Crelox system to assay the function, the consequences of various nonsense mutation leading to different deletion of the CTER. And what we could see is that if we have nonsense mutation that lead to a deletion of 45 or 52 amino acids, then we have nonsense mRNA-mediated decay, and then we have a loss of overall P53 activity and defective cell cycle control. Whereas if we engineer nonsense, if we target in MEFs nonsense mutation that will lead to shorter deletions, then we retain wild type or more than wild type function. So if we compare this conclusion of our mouse experiment with the human P53, what we did is that we looked at the amino acids for which basically if you change one nucleotide in a codon, you will get a stop codon. And we looked at are these mutations found in human cancers or not? Because there's a huge database where you have 20,000 or 30,000 actually mutations of P53 described in human cancers. And here's the answer. All these nonsense mutations have been described in cancer, whereas none of these have been described in human cancer. And this suggests that really the nonsense mutation will have similar consequences in those 2 species. Now, a third argument came from our analysis of, in particular, of discarin and RTAL1 expression in human cells. So discarin is mutated in large fraction of dyskeratosis congenita patient and RTAL1 in large fraction of Oriental-Redarson syndrome cases. And so we compared in MRC5 cells that are P53 proficient cells, and SV40 MRC5 cells that are deficient because of SV40 antigen, T antigen. And you can see here that if you treat those cells with nutlin, you have no effect in the SV40 expression cells, but you reduce expression in the wild-type cells. So this tells you that human P53 is actually regulating DKC1 and ALTA1 as well. Now, a fourth point is given by another gene that has been found mutated recently in telomere syndromes. So this was a 2013 picture of genes mutated in telomere syndromes. If you look at the 2014 picture of genes, then there's another player. It's TPP1. TPP1 was shown mutated in the Oriel-Redarson syndrome in a few cases. So here I'll tell you about some experiments that are unpublished that were mainly done in collaboration and were actually mainly done by David Silvestre in Arturo Londono's lab by studying cells that were prepared by Annelise Benassar-Griseli. So these are human mesenchymal stem cells. And these cells do not express telomerase, and they undergo a replicative senescence. So you can see here that after about 20 passage doubling, you reach a plateau, and they actually have shorter telomeres. They enter into senescence, and you can see TIFs. So this is typical of replicative senescence, and actually if you overexpress H3 in these cells, then you rescue the phenotype. Now, if you look at the senescence in these MSCs, what you can see is that the expression of RAP1 or TIN2 is not altered, but the one of TPP1 is really going down. And you can see the same thing at the protein level. So TPP1 protein levels are really going down. And of course, it's been described for many years that as the cell passages and go into replicative senescence, the P21 levels go up because P53 gets activated. What was really surprising is that if you look at PAT1, you have no change in mRNA levels, but you see that PAT1 is actually accumulating. And it's accumulating in the cytoplasm and not in the nucleus where it's supposed to be. So what's important to consider is that TPP1 and PAT1 actually normally interact together. And if we prevent the expression of TPP1, you can see TIF go up, and you can see PAT1 actually go into the cytoplasm and accumulate into the cytoplasm. So this really suggests that there's a direct connection between decreasing TPP1 level and accumulating PAT1 in the cytoplasm. Now, what's P53 doing in this? If you actually infect the cells with a retrovirus that will allow the expression of E6 protein, that's a viral P53 inhibitor, you will delay replicative senescence. So you will allow the MSC cells to grow for about 8 more passages. So this is classical, this has been described. And you will actually see less PAT1 accumulate in the cytoplasm. And what's interesting is that if you treat the cells with nutlin, you have no effect if the cells are infected with E6, but without infection, without expression of the E6 P53 inhibitor, then you can see that you have a downregulation of TPP1, and you have this downregulation of TPP1 and accumulation of PAT1. So what we think is happening is that as the human mesenchymal stem cells enter into senescence, basically you have activation of P53. This will reduce the level of TPP1 in the cell, and TPP1 and PAT1 need to be complex to actually go into the nucleus, and so you have less and less PAT1 that can go into the nucleus as you have more and more P53 that's activated and that's preventing TPP1 to be expressed. So as you can see here, we saw in the mouse that a P53 activation will act on the regulation of telomere length because it will prevent the expression of ALTA1, or will reduce the expression of ALTA1. It will act on the telomerase by acting on DKC1, and on shelterin by acting on TRF1 and TIN2. And what you can see here is that in human, it's not exactly the same mechanism, but it's basically the same components of the telomere metabolisms that are affected. You have DKC1 and RTL1 that are also downregulated by human P53. And here, shelterin complex is also affected, but this time by acting upon TPP1. So in both species, P53 activation will actually lead to shorter and dysfunctional telomeres. Now, if I had spoken last month, I would have stopped there. But actually, there's another paper that's really interesting that came out 3 weeks ago, and that identified another gene that's important in telomere syndrome. And this gene is PARN. So what is PARN? PARN is a polyA-specific ribonuclease. And it's been shown for many years that actually PARN can actually deadenylate the P53 mRNA, and this deadenylation will lead to the instability of the P53 mRNA. And you can see this here. If you do a siRNA against PARN, you will see that the half-life of the P53 mRNA go up, whereas, for example, beta-actin is not affected. So in this paper, so the team of Dokal identified PARN homozygous mutant, and they compared it to the father, that's heterozygote, and the control, and they found that basically the DNA damage response in this mutant is indeed increased compared to the father or the control. So this is consistent with the fact that PARN actually mutation leads to P53 activation. And what's interesting is that they looked at the levels of various genes that have been shown mutated in telomere syndromes, and what they found is reduced expression in the patient for TRF1, DKC1, RTL1, and TERK. And this is, of course, really interesting for us because out of the 4 genes that they found downregulated, 3 of them were downregulated and are MOS mutant. So this really suggests that there's a lot of things in common between our mouse model and the PARN mutated patients. So this is not a formal demonstration because a PARN could have different targets, but it's the best evidence so far that a mutation that activates P53 may cause telomere syndrome and telomere dysfunction in humans. Actually, I was very fast, and I'll stop there. I'll thank again Eva Simonova for generating actually the P53 delta 31 mouse, and Sarah Geber was also very important in steering this mouse. She's now joined with by Eleanor Toufekjian on this. Boris Bardot and Rachida Bouarish were important for CHIP experiment. Ming Fang on hematopoiesis experiments. Vincent Lejour on molecular biology and mouse genotyping. And so again, Irena Draskovic was important for studying the mouse, like the telomeric aspects of our mouse mutant, and David Silvestre for the human mesenchymal cells. I'd like to thank our sponsor, and more particularly La Ligue and Fondation France. Thank you.